My name is Elena Molokanova. I'm CEO of a company called NeurANO Biosciences. I'm originally from Ukraine, and I am a physicist by training. And I moved from physics to biophysics to neuroscience through studying ion channels in academic and industry. The name of the company NeurANO comes from two words, nano and neuro. So because we're developing nanotherapeutics for neurological disorders, so I thought it was cute. But I think it was a mistake because too many companies start from the neuro and we just got lost. So we sort of think about maybe some more clever names. So just let me stand here so we'll see what so quick outline we're going to talk about. So we focused on disorders with neurologic imbalance, which include autism spectrum disorders. We concentrated on these three different therapeutic indications because NIH decided to support us in these particular areas. Our goal is to develop efficacious and safe drug targeting NMDA receptors. And our solution, as you see, is very different, thinking outside synapses, because many other companies concentrate on making synapses stronger. So we decided to look at from different direction. Now we have a lead therapeutics. I'll tell you what it is. IAM is just like code word to keep you guessing. And we have, again, as I mentioned, funding from NIH, we have IP and running clinical studies, which I'll be sharing with you here. So how many neurological disorders suffer with metabolic dysregulation? I think like a lot, because what I mean, I think actually if we succeed, we're going to be a unicorn company if we're able to do what we promise, because 90% of neurons in the brain have an NMDA receptor. And our drug targeting these particular receptors, and therefore, if we're lucky and if everything goes as we hypothesize it is, I mean, it's going to be huge. So from birth, I mean, children are born during birth, because some trauma, elderly soldiers, sport. I mean, every person can benefit from this disorder. And here, based on this, that's where we're working right now. We have many different disorders. These first three in stage 1, they are further than the others, but everything else is also a possibility in the future. I mean, everyone is asking us when we go, especially for in venture care presentation, how much money can we make? We can make a lot of money. I mean, you can just look at, I mean, you know, not only people experience problems that like to fix, you can also, I mean, earn a lot of money by targeting. So you can look at numbers yourself in billions. I mean, Huntington's disease, which is, you know, rare neurodegenerative disorder, is like still $1.8 billion. But if you go traumatic brain injury, Alzheimer's, it's just like a lot of money. And I'm not even mentioning other indications as they put down there, ischemic stroke, ALS, depression, pain, autistic spectrum disorder. So it's, I mean, kind of unicorn if it works out. Okay, so what NMDA receptors are? As I mentioned, 90% neurons in the brain have NMDA receptors, and glutamate is #1 major excitatory neurotransmitter in the brain. And these receptors take glutamate, open, and allow excitation to proceed throughout the brain. Major role of glutamate is being released as a neurotransmitter, released between two neurons, because it will from presynaptic neuron to postsynaptic neuron, it will transmit information throughout the brain, and the work in physiological normal. When you're thinking, when you're walking, it's actually your brain works 24/7. But if something happened to it, if you have injury or disorder, it means, for example, let's go with again traumatic brain injury. You get hit in the brain, your neurons can disrupt. I mean, just break down like balloon. And glutamate would be released everywhere outside the synapses. And synapses, there are mechanisms to take glutamate back in a neuron and recycle it. Outside synapses, there are no mechanism like that, and it means glutamate will be activating receptors outside synapses, which means it doesn't transmit any information, it just activates this receptor that bring calcium inside cell and kill it. As soon as this next portion of cell died, it again releases extra glutamate. It's like avalanche. That's why traumatic brain injury, for example, people don't die right away. They die, they become much worse over time, because what glutamate does, it just accumulates outside synapses and activate other receptors. So if you take drugs targeting NMDA receptor and decided to use them, you will inevitably block receptors outside and 100% synaptic right here. So if you, for example, ketamine is one of the well-known receptor inhibitors in the receptor. If you take a lot of ketamine, you go on anesthesia. You can, I mean, during war, people taking ketamine is because their limbs amputated without any pain. So ketamine is one of the things. So therefore, you don't want to take drug in high concentration. As soon as you start taking it lower concentration, very low concentration, then it might be not enough of the drug to block bad things that this NMDA receptors are doing. So it's really balanced. Again, if you take small molecule, which people, for some reason, I think it's really incorrect direction of neuroscience drug discovery. Everyone likes oral, therefore they're going for some small molecules. But if you take small molecule, in this particular case, small molecule is so small, it will go inside synaptic cleft and block your thinking, I mean, the anesthesia. You don't want to do that. So therefore, I think focusing on small molecule inside drug discovery may be a mistake in some cases. So this is a picture. So we don't simultaneously cure disease and interrupt brain function. So there has to be a solution around it. So that's what we want to just show you summary of previous slide. This is, you have a synaptic cleft, and we would like to spare NMDA receptor located in synapses, but block all receptor located outside. Clearly, small molecule won't be able to achieve this goal. So existing drug, so for example, memantine, it's a FDA-approved drug, Namenda for Alzheimer. So it's an NMDA receptor inhibitor. But since it's a small molecule, it blocks 60% of receptor outside and 40% of your synapses. Therefore, you have to take it at low concentration, and you have to take it only in moderate to severe Alzheimer. When you have fewer synapses, they might be blocked and affect normal working relations. So far, it's clearly not optimal. So being a physicist, as I mentioned, I approach the thing from an engineering standpoint, right? And we just thought simple things. So it's dimensions of synaptic cleft is about 20, 25 nanometers. If you will design a drug that is larger than synaptic cleft, then you can, I mean, you just won't be able to get there you don't want it to be. And it might work exactly how we want it to be. So we'll be able to avoid blocking synaptic NMDA receptor and block only the synaptic receptor as was shown on previous slide. So idea would be that we'll need to bulk it up somehow, but use pharmacological warhead, preferably already approved by FDA. So we don't want to fight another battle trying to show that our pharmaceutical works. And advantages, we can apply it at any dosage without worrying that we might block normal brain activity. We can apply it, use it at any disease stage because we don't worry about having too many synapses in your brain, which is actually good for you. So many advantages to using this approach. So this specifics how decided we go about it. We just run incarnation, yeah. But at the end, in the center, we have a central small gold nanoparticle. It doesn't have to be gold, but gold allows us to use a lot of known about chemistry of gold, how to conjugate, how to make it stable, how to make it exactly 20 nanometer, 10 nanometer, 15 nanometer if you want. So we have really strict control about size. And now drugs is about size. And then we fitted it with two different packs for different purposes. But at the end, we have a conjugated molecule, in this case, memantine. And this molecule is large enough to stay outside synaptic cleft, but still small enough to diffuse sufficiently throughout the brain because we know what the size of a pore in hidden cells in the brain. There is a positive things. It's an act on clinical valid therapeutic target. It's clinical valid therapeutic target because memantine is approved for Alzheimer's. So this is sufficient as it is. Uses validated FDA-approved warhead. Can have a favorable safety tolerability profile because we don't cause all the side effects that broad and NMDA receptor agonists do, because we avoid synaptic cleft. IC50 show in nanomolar range, show 5 nanomolar in the particle units, and reduced dosage and adherence to professional compliance. So as I mentioned, I don't like small molecules because they have many problems, but large entity obviously have many problems. So number one is how do you live into the brain? and how it will diffuse throughout the brain. So therefore, before telling you about our preclinical data, I want to assure you that we look at this very carefully. So there are several methods to purposefully deliver nanotherapeutics of that size. And I mean, you know, the pickle is used for many cases. We don't even have to consider application like, for example, for Huntington. It's known that one of the famous companies is doing 12-hour brain surgery by drilling hole in the brain and delivering the gene therapies inside brain therapy. So we don't even have to go to that option, but if needed, I mean, theoretically we can do it. But after you deliver it to the brain, the question is it's going to be staying right where it was delivered, going to be diffusing through the brain. And again, many papers look at it at now. I mean, Vice President, the Nanotherapeutics, he studied extensively which kind of nanoparticle can go through the brain, and it's known this is a dimension of the spaces in brain parenchyma, at least 38 nanometers, which means they are larger than all nanoparticles, and it means they will be able to diffuse freely. So this is again blow up of the same figure, so you will see what it is, and this is the trajectory showing, I mean, how fast particle 40 nanometer, we have 30-35 can diffuse, and on the right you have a paper showing dimensions of a force in the brain, and you can see up to you go to from 0 to 25, 26 to 50, you have more than 75% covered by the size range. Yeah, and here we have shown if you use brain slices and based on the particular drug, you can see that diffusion coefficient is similar to dextran, I think, sizes like 40 kilodaltons. So it's very easy. And if you inject it into the brain and just look at one particular area of the brain and just look at the diffusion over different time points, you can see that stabilizes for about 4 weeks. And here you can see 3 months, different time point. They have taken brain slices, and this is a gold nanoparticle. They can reflect light. You can use a dark field microscopy to see these nanoparticles in the brain. And you can see they're homogeneously distributed as like a small light bulb. So I mean, we know we can deliver it in the brain, and it stays in the brain for a long period of time, which is really unusual for drugs. It's more closely to gene therapy thing, and you can do it once and just see the lifelong effect. But it's definitely much better for many disorders where like lifelong suffering, like Alzheimer or even autistic spectrum disorder. So our number one indication right now is opioid use disorders. I mean, everybody knows it's a huge pandemic, and as we saw from many movies, I didn't experience myself or anyone with this problem, but in many cases, people acquire opioid dependence because they were given bad advice in using opioid the drugs for pain. And right now it's a problem how you can deal with it. So as I mentioned, there are several stages: withdrawal, development of opioid dependence, and relapse. Here we started with looking at the Goldman-Martin effect on withdrawal, and really just scheme how it works if you want to go through this, but I mean, just standard one. You train mice to recognize different chambers with different orientation, and then give them drug in one of the chamber, and they'll remember whether they like it or not. And the presence of the drug, how they're gonna do. So we can show that we can, actually, when people try to stop being opioid dependent, withdrawal is the most severe process that prevents them from doing so. Therefore, helping them to go through the things so they can take, I mean, some of the drugs available only when you were without drug for 2 weeks, and many people cannot, and just the 2 weeks is impossible. But this particular case, we didn't see any withdrawal for the mice when they were given drugs simultaneously with morphine. And this is things that I mentioned that we can do intranasal, because as you know, a lot of drugs for opioid use disorder are given intranasally to stop. And therefore we have, and there's a reason for that. You can see when you go for nose, all these areas labeled like olfactory neurons and the PVN and the area, it's where actually opioid dependence happens. So for drug to travel from nose, from olfactory pathway to the area, it's very short distance. So therefore, it's really appropriate to use it for the things. So we have given mice twice infusion of globin saline and repeated the same chemical paradigm, and we have shown that we can deliver drug its concentration sufficient to have a withdrawal even through intranasal delivery, which is great. It clearly requires optimization because in this particular case, device that we use for mice definitely not the device used for humans, and there are many different devices for humans. We have to play around with it, which is required for during clinical phase trial. You have devices that, passive, active, with gas, all the different things. And right now, Aptar is a company that's developing these devices. It's actually look pretty much like a device you guys use when you have a nasal congestion, but the insides look different. And right now, Aptar is developing so-called nose-to-brain delivery device that allows you, instead of just spraying your entire nasal cavity, you just allow to forward spray of the drug toward upper roof of the nasal cavity, very close where olfactory pathway is. So hopefully we're going to be benefiting from this discovery, just to have more efficient delivery of the drug, because it's still going to be delivered, but just sufficient in high quantities. Here is things that might be interested all of you in the future. Because I know my husband had a surgery, real complicated. He was given opioids and drugs that should go with opioids to make sure he won't overdose. It means that, people can develop opioid abuse dependence just by taking opioid medicine in several cases. So here we have shown, right now, we repeated it like 4 times, different configurations. We know that if animals are given our drug before giving morphine, they do not develop opioid dependence at all. 0. So which is, incredible thing, because, for people, for healthy people, for people who don't want to get opioid use disorder, for people, I think it's really excited when they receive this, obtain these results. So I have more data, but I just want to get you excited. So second is Alzheimer, because Alzheimer is I don't have to tell you how bad Alzheimer is. And recently, luckily, we got a couple of drugs approved, a couple of therapies approved, which people started believing that it's possible to develop drug for Alzheimer. They are not as good as everyone hoped them to be, if only because even if you will reduce, they're anti-amyloid drugs. So even if you remove amyloid from the brain, it's not clear whether there's going to be effect of cognitive behavior or any other thing, because the biomarker was way you able to do that or not. That's why they were approved. And many side effects that are not good. So in our particular case, again, Alzheimer is a neurodegenerative disorder. It means neurons are dying, they're releasing glutamate, you get overexcitability, you have excitotoxicity. So very, very simple thing that guarantees that could have happened and definitely happened in neuroprotection. Because you're protecting, you're closing down these receptors that are not located synaptically and not allowing glutamate to cleave cells. So it is expected, and we received this data. But most important thing, and very cool things that we have also found, is that these 2 graphs on the right side, actually 3 ones. So you can, in the middle, there is a BDNF. It's a very important factor in the brain. So for example, UCSD is going through clinical trials when they inject BDNF with virus inside people's brain because they believe it's going to cure Alzheimer on its own. In our case, by just using our drug to provide neuroprotection, we're able to dramatically increase BDNF production on its own in the brain without doing virus delivery to the brain. 2 graphs on the right is we have shown that we can reduce number of amyloid aggregates in their sizes. Again, that's what anti-amyloid drugs are for. But we have found going through longer pathway, we're able to achieve pretty much the same thing. I have to make sure that it's obviously in animals. And many people got excited with the results in animals. It's a very well-known 5 FXAD mice. It's familiar Alzheimer disorder mice with 5 known mutations. But so far in this particular model, it looks terrific. We'll see how it goes further. We're just compiling them and submitting phase 2 grant on the of aging. But I think it's very exciting that they're not able to provide neuroprotection, but hit to other points that other drugs trying to achieve. And also Huntington, because I mean, social neurodegenerative disorder, many mechanisms are similar with Alzheimer, and people are running clinical trials with memantine as well. They were inconclusive. I mean, one was better conclusive, two others not. And I think it's possible because memantine was applied used on low concentration and they're not given its ability to show its full force. So we have done some study in Huntington, and it also looks promising Huntington's disease. So we're going to be pursuing this. All the studies are done in collaboration. So it looks like, I mean, it's a startup, a small company. All the studies were done in collaboration. So I mean, I wanted to receive this result, but these results are received independently in academic labs in DC, in DC Irvine, in world-class professors that work in this particular area. And this was a small study, but I think it's also because, I mean, traumatic brain injury had the highest dollar sign assigned for profit. We have done only small study showing that if you will take animal and give it, hit it on the head with a hammer, it was like modified scientific hammer, but still in 24 hours you give it drug, you can reduce, you can improve its cognitive performance of the mice and by day 5, which is extremely important. Because let me make sure that here we have done drugs, we have given drug 24 hours of traumatic brain injury, but obviously the sooner you can give it, the high effect you might get. But again, so far we have our hands full with 3 other projects, but I think this one is really exciting, just waiting for us to free up time to work on something like that, maybe find collaborators who would be interested in that. So yeah, I mean, it's like from my other grant, we have done quite a lot of different. So we tested different. So I didn't mention here that we have also tested our drug in one of the mouse models of autistic spectrum disorder, and it works there as well. But again, we have our hands full with other things, and we have done some interesting things. I think we are passed into greater things. Yeah, it's our team, my collaborators. Yeah, I'm ready for questions.
Question.
So we try 2 different concentrations, and so far the longest study we have done in Alzheimer's disease mice. In this case, we decided instead of focusing on concentration, try to determine which time point in disease progression would be more critical. Because I mean, we can always, I mean, with mice models, we know they're going to get Alzheimer's, so we can give them drug, let's say, 2 months old. But in reality, people, I mean, we cannot do that. So we have done 3 different time points, and 2 months old, 6-month-old administration, 10 months old, and comparing how less or more efficient drug would be. But we have, and those depends because of upper concentration, and in phase 2, which can be like 5 times more money, we're gonna obviously run the entire target concentration, minimum dose, highest dose, whatever it's gonna be.
So some of the best data I've seen on like the synaptic versus extrasynaptic NMDA receptor work has been in stroke, where there's some really old, elegant studies. I used to have the papers memorized, but it looks like you have your hands full.
Yes.
But it seems like stroke might be a good option to pursue. You could do the intrathecal. It would be a justification for like an acute intrathecal delivery.
Yeah, it's possible. I mean, we can, yeah. I mean, we just hands full. I think it's just explanation, right? And we have right now full obligation on 3 grams that we have, and yeah, it's a possibility. And we have started thinking maybe our 4th application would be pain, because of this ability to prevent opioid occurrence from happening. It might be just playing with this pain pathway. And I just show you a little bit of data, but we have some data showing maybe they interfere with pain above certain threshold, and we just do it on that. But yeah, definitely ischemic stroke is, I mean, traumatic brain injury is the same. It's like, it was well known for, I don't know, 30 years that it works this way, but it just, yeah. But I think if people already collaborate with us, we can divide and conquer. And
yeah, great presentation, Alina. So I had a question about dose as well. For the autism project you discussed, how much was the difference from ICV versus intranasal? And second question following to that is, do you also need to show the dose receptor response in the nasal mucosa or in the brain, demonstrating that is there really a good bioavailability of the drug in the brain?
So again, we haven't optimized it much because I mean, we have a company here, IRS, if you've heard about, is a guy who developed the nasal delivery of epinephrine. It was approved recently, and just yesterday I read that some of the company, I think in New Zealand, bought rights to market their drug for $150, just for $150 million. I'm sorry. So that means, I mean, it's getting recognized, right? But still, a lot of people have doubts about nasal delivery, therefore not sufficient efforts. So if we'll decide to go that way and decided, I mean, to work that way, probably gonna apply for a separate grant because, I mean, we have many different excipients to try, different concentration. So we apply drugs. Usually we put, when there's the feed delivery, we use, I think, 5 microliters because, I mean, of limited volume of CSF. In case of nasal delivery, you can do it longer, you can do it in multiple dosage. I'm not remember, I think here we use, I think, 20 microliters, like in 5 microliter space apart to just wait. And we haven't optimized it pretty much at all, which was surprising because, I mean, this was from very first attempt. And I mean, we guess we read literature, we try to put some stuff, but we haven't done any optimization. So I think it's highly, highly beneficial in the future. I mean, so.
Thank you.
Any other questions? Okay, thank you very much, Dr. Elena Molokanova.